Hello, my name is Colin Martin, and I would like to welcome you to another video for MCDB 427, which is Molecular Biology at the University of Michigan. Over the next few minutes, we're going to be discussing figures 13.15 through 13.17 from the book Molecular Biology, which is written by Robert F. Weaver. And these figures are going to help us get a better understanding of nucleosomes, their function in preventing transcription, and ultimately the main topic for today, nucleosome-free zones, how they can be located, and how they affect transcription. Now I want to take just a brief moment to review some basics that you might have already covered in another intro bio class regarding the structure of chromatin. And each of these individual units that we see here are what we call nucleosomes. Each nucleosome is composed of DNA that, when wrapped around proteins called histones, which are colored in blue in this figure, make the DNA mostly inaccessible to RNA polymerase, which inactivates transcription. Now, in order for the DNA to be transcribed, RNA polymerase needs to have a way of gaining access to the promoter of the sequence of interest, and that can only happen when nucleosomes are not associated with that particular region. So essentially, in regard to nucleosome position in chromatin, there are two basic scenarios in which transcription can be initiated. And the first is when a nucleosome happens to be covering the promoter region on a sequence of DNA which blocks transcription from occurring, as it would in this case. A number of chromatin remodeling factors can move these nucleosomes out of the way, however, which would then allow RNA polymerase to activate transcription, which is initiated here as an arrow at the +1 site. The second scenario involves a sequence of DNA whose promoter region is not covered by a nucleosome. And in this case, an activator and any other associated factors can bind the DNA directly and recruit RNA polymerase to immediately begin transcribing the gene. Now, a man named Moshe Yaniv wanted to investigate these relationships further, specifically with regions called nucleosome-free zones. He and his colleagues wanted to determine what kind of evidence they could gather about their existence in the control regions of genes that were able to be transcribed. So they asked themselves, is it true that genes whose control regions are not complexed with nucleosomes are transcribed? Are there nucleosome-free regions that we can detect? Well, the first step towards figuring those things out was locating where exactly these nucleosome-free zones were on a sequence of DNA. So Yaniv chose to conduct his experiments using the very well-known SV40 viral DNA. And there were a number of reasons why Yaniv chose to use this specific DNA. And its relatively small size and the depth of knowledge that had been published about its structure were extremely helpful. But another important reason it was chosen was because once SV40 is infected into mammalian cells, and in fact it was monkey kidney cells in this experiment, the DNA forms complexes of nucleosomes that are very similar to our own cellular chromatin. This is where the name mini chromosome comes from, and you'll hear me refer to them as such throughout the video. So one of the first steps Yaniv and his colleagues took was to visualize the SV40 DNA directly using electron microscopy to determine if any nucleosome-free zones existed in its structure at all, and if they did, their position along the strand. And these are the images that resulted. Now, just to orient you, what we're looking at here are two populations of SV40 mini-chromosomes, the first being in panel A, and a second population in panels B through D. Each of the mini-chromosomes is a circular strand of DNA with a length of 5,224 nucleotides, and each of them has about 24 nucleosomes complexed within its structure. You'll notice here in part A of this figure that these particular mini-chromosomes have a very consistent structure throughout, and I've drawn a cartoon version of what you're looking at to give you some other form of reference. Each of the nucleosomes, which are these bigger dots here, is spaced out relatively evenly. So over the 5,224 nucleotide sequence, we're expecting to see a nucleosome every 217 base pairs. These four figures, on the other hand, look a little different, and each of these mini-chromosomes, which are still the SV40 mini-chromosomes Yaniv was investigating, have a stretch that doesn't have any nucleosomes at all, despite having no difference in the total number of nucleosomes present. And this happened to be occurring at approximately 15 to 20% of all SV40 mini-chromosomes that were infected into the monkey kidney cells. Again, you'll notice on this side of the mini-chromosome here that there are a significant number of nucleosomes, but there's a stretch on the opposite side of, on average, 248 base pairs that looks totally bare, and this is the nucleosome-free zone. Now, one of the other big reasons Yaniv decided to use the SV40 viral DNA was because previous experiments had shown that treatment of the mini chromosome with a restriction enzyme called Bagel1 makes a cut in the control region for a number of different transcripts that we see here in this image, which I've included to give you a more detailed look at the features of SV40. And we'll use both and compare exactly what we're looking at. It became of interest to them to see if treatment with the restriction enzymes Bagel1, BAMH1, or EcoR1 would lead to any definitive conclusions about the location of the nucleosome-free zone. If Bagel1 cuts in the control region for the early and late genes in this sequence, and if we could determine that this cut was being made in the nucleosome-free zone, then it's certainly possible that this region, being devoid of nucleosomes, is no accident at all. A nucleosome-free zone in this region could mean all of the necessary sequences for transcription, including the promoters, would be available for binding by other transcription factors and RNA polymerase to induce transcription of these genes here that I mentioned earlier. Yaniv and his colleagues needed a way to reliably test these hypotheses, but there was still one issue going forward with the experiments, and that was the fact that SV40 mini chromosome that he was investigating was circular. And to test you a little bit here, I'll pose this question to you. If you were conducting an experiment like this, and you wanted to identify exactly where the nucleosome-free zone was on SV40, why would having circular DNA make your job more challenging? Take a moment to think about it, pause the video, and see if you can come up with a good reason. Well, if you've had a chance to think about it, you may have discerned that without any way of identifying any landmarks, it would be much more challenging to figure out where along this circular mini chromosome you are at a given time. It's important to note that treatment of the mini chromosome with a single restriction enzyme would not be sufficient for determining the location of the nucleosome-free zone. So what Yaniv decided he would do was use high quantities of restriction enzyme to cut the sequence at known positions using Bagel1, BAMH1, or EcoR1. And because Yaniv knew the sequence of SV40, he could draw himself a map of where he believed the nucleosome-free region to be located with respect to these restriction sites. Figure 13.16a on top here shows the locations of each restriction site along the mini chromosome. And part of the genius of this experiment comes from the fact that Yaniv chose these specific restriction enzymes to conduct his experiment. Notice that the Bagel1 site is located near the control region of SV40 here on the lower left. Imagine how challenging it might be for RNA polymerase to conduct transcription if there were nucleosomes bound around this region of the DNA. It shouldn't sound like a totally radical idea then that Yaniv would propose that of all places along the SV40 mini chromosome, that if transcription of the genes would ever take place, that this would be the location of the nucleosome-free region. And again, this was just a hypothesis at this point, but by picking a restriction enzyme that would cut near one of the edges of this region, Yaniv could experimentally confirm or deny without a doubt whether or not this is where the nucleosome-free region was located using microscopy after treatment of the mini chromosome with restriction enzyme. Figure 13.16b shows what Yaniv expected the products of each restriction cut to be. If Bagel1 cut within the nucleosome-free region as he expected, the result would be a long sequence of DNA bound by nucleosomes with a tail that had no nucleosomes associated with it at all. A cut with either BAMH1 or EcoR1, on the other hand, would result in a nucleosome-free region flanked by two regions of DNA bound by nucleosomes. And, as it turns out, this is exactly what Yaniv and his colleagues observed. Figure 13.17 is direct evidence of the fact that cutting with BAMH1, which is shown in these first three images, results in the nucleosome-free region located in between two nucleosome-rich regions. Cutting with Bagel1, on the other hand, shown in the final three images here, shows us exactly what we expected: a long stretch of DNA bound by nucleosomes with a tail that is nucleosome-free. I hope that you found this video helpful. I wish you the best of luck in your studies, and as always, go blue.